in the Department of Bioengineering. He did his PhD in bioengineering from UC Berkeley and San Francisco before going on to conduct his postdoctoral studies at the Center for Engineering and Medicine at Harvard Medical School and Massachusetts General Hospital. The research in his lab takes advantage of unique physics, microenvironment control, and potential for automation to develop miniaturized systems for applications in medical diagnostics and basic biology. Today, he'll be talking about some of the latest developments from his lab and speaking to the rich entrepreneurial spirit and the ecosystem that we have here at UCLA. So, still loading the presentation.
Now you get put on the spot. What else do you know?
Okay, and Dr. Carlo's talk is titled Commercialization of Bioengineering Technologies from UCLA.
Can you hear me in the back there? Thanks, Dan, for the introduction. Thank you all, prospective students, graduate students. And we also have some high school students in the back. So a lot of generations and a lot of different audiences to speak to. So hopefully I can speak to all those audiences in one talk. But I wanted to share kind of as Diane mentioned, this ecosystem of entrepreneurism and translation that we have here at UCLA and really highlight a number of companies and startups that have come out of the different labs in bioengineering. And then I'll go into a few vignettes of work that's come out of our lab. And hopefully that gives you a sense of some of the things that are happening at UCLA and how we are taking advantage of that interface between engineering and medicine across the street. So we have a lot of startups at UCLA out of bioengineering. So I had not known that we had this many startups until I started collecting this list a few weeks ago. And they cover a wide variety of spaces. So we have diagnostic startups that cover from all the way from nucleic acid diagnostics to analyzing cells to point of care diagnostics to ways to analyze the immune system earlier and also use some of our consumer electronics technology like cell phones to make diagnostics cheaper and communicate widely diagnostic results. So really amazing set of work that's coming out. And this is our department here, a little over 10 years old. Therapeutics, you heard some of the great work that Dr. Zhen Gu is conducting. We have other work that's working on vascular grafts and other technologies in Suna, another company called Obsidio Medical, and a number of therapeutic technologies that are being developed. And also medical devices. And I'll talk about one of these vignettes from a company called Tempo Therapeutics, which is building off of some microgel technology out of our lab. And then finally, we also developed a number of automation tools or tools that can be used to enhance or accelerate life science research. And so that's critical as we try to increase the quantitative data sets that we start to develop in life sciences and use that to enhance all these other areas of therapeutics development, medical devices development. So these technologies are also, these startups are also along the various routes of commercialization and have made really significant milestones towards getting the technologies that they're working on out into the world, right? So starting a company and creating a nice logo is the first step. But obviously that's not the last step in commercialization. And some of the logos, if you look at some of the early logos like Amazon, others, they're not great logos. So maybe the logo is not as important as actually what is done by the people that are part of these startups, right? But you can see that there's a number of startups that really are moving down this progress towards translation. Of course, you're going to see different stages because medical devices and therapeutics are going to take much longer to get into the clinic, obviously, than a new research tool, which can be used by scientists much earlier. It doesn't have to be perfect. It doesn't have to have regulatory approval. But you see, companies like Omega Biosystems, which was acquired by Becton Dickinson, came out of Bahram Jalali's lab here at UCLA, and is commercializing new flow cytometry technology. And I'll talk about temporal a bit later, but I'm proud to say that they submitted their first de novo 510K package to the FDA for their product to be approved in a wound healing application. Of course, it's not approved yet, it's being reviewed, but that's the first step. And you can see that these companies are along at, some of these other companies that are developing diagnostics and other regulated products are, in various stages of fundraising and bringing people in to make these technologies that really started out as ideas in the lab from people like you into real products, right? And that takes teams, it takes people with different expertise. And so many of these companies are now building those teams. And this has really been assisted at UCLA by this ecosystem that is promoting entrepreneurial activity. And there's a number of components to that ecosystem. I'm highlighting a few of them. So we have our technology development group at UCLA. This is the group that works with students and investigators to file patents, to license technologies, to teach students about various aspects of business formation, for example, through seminars, through interactions. They help find investors. So this is a group that's really been critical for a lot of the startups that come out of UCLA, and of course is important because of how they interface with the intellectual property that's developed here at UCLA. We also have incubators, for example, Magnify, which is an incubator that's located at the California Nanosystems Institute. So California Nanosystems Institute is this institute was originally funded by the California state government, where this was to form interfaces between academic environment and industry. And so here on campus, we have a building and we have facilities that are at this sort of interface, including incubator space. And we have a number of our companies that are associated with this incubator space called Magnify. So they provide these companies a space, at early stages when they don't have much money to rent out huge office spaces, but then also access to facilities that can be important for those companies to move quickly without having to spend a lot of capital on buying new large equipment, for example. We also have other incubators in the LA area. So LA Biomed is one that several of the companies that come out of our program have been associated with. This is down in the Torrance area, and this is also associated with Harbor UCLA, so other healthcare systems that are connected with the greater UCLA health system. As I mentioned, UCLA Health, that's a critical component of this ecosystem, which without, you really don't have that interface to what the clinical needs are, what are the important problems that we need to be solving, and how can we test those problems and really iterate and vet whether these technologies will work with the patient population. A few other institutes that we interact with are the California Center for Accelerated Innovation, which funds technology at an early stage that needs to maybe a little bit more funding to get to a commercial level of readiness. CTSI also helps with some of this, and Technology Development Group also has an innovation fund. Well, they'll fund up to 100 to 200,000 to further develop technologies to the next level before startup formation or licensing. So a lot of resources on campus that help to create this ecosystem and go from paper and patents maybe to now products and impact with patients. So I want to start with one vignette from the lab that's an early stage startup called Foresight Bio Technologies. So there's a lot of puns in this name of this. So the idea here is that we're measuring the force that cells apply as a marker of disease. And the student that really took the lead on this is Dr. Ivan Pushkarsky. He was a PhD student in my lab and is now the president and was the founder and is the president of Foresight. So the problem that we're trying to solve is that there are not good ways to develop drugs that target contractility of cells. And why was that important? Because contractility of cells is integral to a number of disease processes and almost all of our tissues, right? So like obviously our heart, skeletal muscle, but a number of smooth muscle tissues, which if there's problems with those tissues, that leads to disease states. And I'll give you one example. So your airway smooth muscle cells, right? If they contract too much when exposed to an agonist, that leads to asthma. So I was personally very familiar with this because my son, when he was, we just moved back to LA, 5 years old, we'd wake up in the middle of the night and he couldn't breathe, right? He had an illness, you had a cold that leads to triggering of asthma, so it's called reactive airway disease. And we're going to the emergency room, right? You can't. You're a 5-year-old or you're a 3-year-old can't breathe. You're gonna go to the emergency room, and what do they do for them? They're going to provide either steroids. That's all they can do right now to help open the airways. And that's not great. But what I found out is actually there's not many treatments that are available for people that have asthma. Right? So you have beta-adrenergic receptor agonists, beta agonists that are an inhaler like albuterol that you're probably all familiar with. So those are short-acting beta agonists. And then you have long-acting beta agonists, which are supposed to get released over longer periods of time. But those kind of compete with each other, and so there's really only one class of drug that acts to open up these airway smooth muscle cells. There are other drugs that target inflammation, but really there's some missing pieces in this scenario. And so this is where we first kind of started to think about this problem of contractility and connecting it to a particular disease, in this case asthma. But of course this covers a number of other disease states. Intestinal dysmotility or dysmotility disorders where you don't have contractility of intestinal smooth muscle cells. You have preterm labor where you have over-contractility of uterine smooth muscle cells. You have a number of issues with like incontinence where you have bladder smooth muscle cells and other cells that are acting in a way that is not the optimal level of contractility. Okay, so the sum of this is that contractility or force generation within cells is super important. But we don't have tools, especially high throughput tools, to measure contractility of cells. And why do we need these high throughput tools? So we can screen new drugs that can target them. Okay, so that's what Foresight is working on. And their approach is called the FLEX system, another fun name. Fluorescently labeled elastomeric contractible surfaces. So flex is a bit easier to say. And basically it's a simple idea, and that's why I was so excited about this idea. You just embed protein patterns that have some fluorescent molecule that you can easily see into an elastomeric layer. When a cell adheres to that fluorescent pattern and it applies force, it shrinks the pattern. By shrinking the pattern, you can get a quantitative readout of how much force the cell is applying. Super simple. But you can repeat this process over and over thousands, tens of thousands, hundreds of thousands of cells, and each time you can get basically the same quality measurement. Of course, there's some technical details of how do you pattern these proteins into these soft materials in a repeatable way. You can't just stamping onto a really soft thing is not really easy to do. So there's some details of that, but we were able to achieve it. So this is kind of a picture of a cell that was adhering to one of these patterns and then contracted that pattern. And you can see this pattern had no cell attached to it, and a very clear difference in the deformed shape of the pattern, which can tell us more about how much force that cell is applying. So how can we use this? So we did an experiment which I like to call asthma attack on a chip. We start with cells in 96 well plates that are seeded onto these patterns. So single cells on these patterns. We then expose to an agonist. This is bradykinin, 10 micromolar bradykinin. We then image. We then give the either inhaler, formoterol, or we don't give an inhaler, and then we image again. Inhaler drug. We didn't actually give the cells an inhaler. That wouldn't make sense. And so what did we see? Okay, so we did this with patients that were healthy patients. We didn't do it with patients. We did it with the cells extracted from healthy patients that died for other reasons. And we did it for patients that died because of fatal asthmatic. Okay. And so this is a histogram of many single cells and how much they displace the pattern. And as we give bradykinin, this is the asthma attack starting to come on, right? And then as we give the albuterol or formoterol, we see that, okay, we can start to relax those smooth muscle cells again. And this is the types of patterns you get from 6 different patient cell lines. So these are thousands of single cells for each of these. And if you take the average, then you get these particular plots here. And you can see, okay, here's the asthma attack coming on, and then the relaxation. You can see there's some interesting things. They all behave relatively similarly, but there are unique differences between patients, right? So there's some aspect of personalization that might be important here as well. And we know that everyone will have slightly different responses to drugs as well. One thing you see is in the fatal asthmatics that they initially have higher tonic contraction. The rate at which they increase is actually similar to healthy patients. And the rate at which they respond was slightly different, although you can't see that with this data, but it's slightly different to how they respond with the model. Okay. So this is one particular example. And we can also do similar types of experiments. This is showing a time-dependent experiment where we're looking at cardiomyocytes that are contracting patterns, and then we're looking at the force plots, or the deformation plots for cardiomyocytes. You can see these kind of different types of signatures. Here's where we are actually pacing the cardiomyocytes. These are heart cells, obviously. And you can see some interesting things. So here's the pacing at 1 hertz, and then we start to pace at 2 hertz. And you can see that they don't have time to relax fully at 2 hertz, and then pacing at 1 hertz and 2 hertz, et cetera. So the next step is can we use this type of platform to screen for new drugs that can target contractility? So this is a proof of concept for a 384 well plate. So this is standard plates that are used for laboratory automation. And what we see in this 384 well plate, each well has thousands of these micropatterns, and then we have hundreds of single cells in each of these wells. And from each of these wells, we give a different drug. And so for each well that had a different drug, we get a point, which is the median contraction of all the single cells in that well. And then we can find, this is kind of the average, and green is a control. And so we look at some number of standard deviations from this control, and we find hits. So compounds that relax these cells beyond what we expect from noise. And so that can allow us to then identify whether these compounds are unique that could be acting through different pathways to treat this type of disease process. And this case, we looked at, this is called LOPAC, or Library of Publicly Available Compounds. So these are actually compounds that are already approved drugs in most cases. And so if they are different and can be used, again, for these patients, there's a much faster path to approval because they already have well-known profiles of safety and toxicity, for example. So Foresight is continuing with commercialization of this technology, working with pharma partners to screen not just airway cells, but a number of different types of cells, and also doing other types of development efforts. So the last second vignette is I'm going to talk about an approach that was developed in our lab by a postdoc, or it was a PhD student, Westbrook Weaver, along with a postdoc in Tatiana Segura's lab, Don Griffin. And so you heard of maybe a little bit about this morning from Jake Kuhn. But the idea is that if we want to create a regenerative injectable scaffold, we need to think about creating microporosity in that scaffold. And we want the scaffold to kind of seamlessly integrate with the surrounding tissue so that we can get tissue to grow immediately into the scaffold immediately after we inject it. Okay? And so the simple idea here is to use a slurry of spherical microgel particles, flow that into a wound, and then anneal those particles together so that they start to stick to each other and to the surrounding wound bed. That porosity is then formed within the negative space between these spherical microgels, and tissue can integrate into that microporous structure immediately after delivery. There's no degradation of the biomaterial that's needed. There's no activation or other processes. The cells can migrate at time zero. So we make these gels, as you saw maybe from Jake Hume's presentation, using microfluidics. So in this case, we had two parts to the gel, a cross-linker part that was sensitive to matrix metalloproteases, and so could be degraded by the body, so that the final gels could be degraded by the body, and a 4-on PEG vinyl silicone component. Those get mixed together. They then form droplets, and those droplets cure over time and to form microgels. Those microgels can then be purified and put into a slurry form for use. And so this is kind of the use case, and you saw the video also probably from Jake Yoon, where you have a syringe that includes these gel particles. You have a wound process in seconds. You can fill that wound. You then anneal these particles together using either enzymes or light or other techniques. You have large pores that form, and tissue can grow in, and then the gel is resorbed so that you have regenerative healing process. So this is some of the images from Jake Jung's work, where you can see cells growing all through these types of particles in vitro. And I want to share some data from Tempo Therapeutics, which is commercializing this in a wound healing application. Oh, I forgot to mention, if you're squeamish, look away. You already looked. So now you know what you might be looking for if you were squeamish, so you can look away. Okay, so they're using this in, as I said, they submitted the FDA package, but in the meantime, they've been working with veterinarians, and these are cases that are referred for amputation. So these dogs would have limbs amputated because of sarcomas or other, like a coyote attacked the dog, different reasons. And so this is what you see after the surgery. This is Mocha. Mocha had a wound here after a soft tissue sarcoma was removed. We put the MAP gel in this wound, and you see this process that basically a granulation bed forms very rapidly, and that granulation bed then heals in a very regenerative way, where you see hair follicles forming over this wound. So this is a wound that would not heal normally in a way that would be satisfactory, and that's why they're referred to amputation. Here's another case of Misty. So Misty had a hemangioma. The reason why these dogs are referred to amputation is because often after the resection of the tumor, you have exposed bone, tendon, fascia, things that are really hard to heal over. And so in this case, we also put the MAP gel matrix into these wounds. You form this granulation bed, and you have regenerative healing with hair follicle formation. And hair basically filling over where the wound was. And there's a third case of Charlie. So this is Charlie. Charlie's ears. Ear was damaged during a dog fight. And there's exposed cartilage in this ear. Again, we used the MAP gel system here, and we see a quite good healing of the over this exposed cartilage region, which is extremely difficult to heal. Okay, so let me conclude. I think I've been too long, actually, but since I'm the last speaker, I can do that. So I think some key messages is that translation is really embraced here at UCLA. So, beyond the papers, publishing papers, we're asking the question at early stages, how can this work benefit patients and the healthcare system? How can we have that impact that's beyond that publication? We have this entrepreneurial mindset where we want to take the next step in development of our technologies, knowing that if we just leave it for someone else to take the reins, that's not likely to happen in many cases. And if we really believe that this is something that's useful and going to make an impact, that we should be taking the next step with. And that we build bridges between engineering and medicine. Really, engineers and doctors have a lot of common goals. We want to solve problems. I think basic scientists have different questions, biological questions that they want to ask. There's curiosity-driven work, which is great. I'm not saying that that's bad, but I think that doctors and engineers have a lot of common goals in how we solve problems, and I think that that's something that we take advantage of. And so you heard this already this morning from the mayor's office, that UCLA is actually first amongst all universities in performance and startups. The number of startups that UCLA creates is more than any university, like you said, not just in California, but all the US. And I don't know if they did across the world. I think it's just only in the US that the study was conducted. So there's this, what that tells you is that there is a willingness of the students, the faculty, and the environment to take those risks to bring things to the next level. Okay. With that, I'll thank you all for your time and open maybe for a few questions. Thank you.
But how far away would that be from trying it in human tissue?
Yeah, so you got to talk to the FDA. It's in their hands. So we've had several conversations with the FDA already, and we've done the studies that they seem to think are necessary to show that it's a safe product. But we're going to hear back from them shortly this year, and we'll know what more things we need to do, if there is anything more we need to do, and then it will be approved to be used in humans.
How common is it for researchers who go on to start companies to continue to do their research on campus?
Oh, so like in an incubator on campus?
Or in the labs on campus?
So yeah, so there's always this interest, this issue of conflict of interest, where if there's a company that's started, that cannot make use of university property, basically. So that company has to pay for using university facilities separately. So they can't be in a lab once a company has started, but they can be on campus. So like these incubators allow for that. And then they can also use facilities on campus as long as they pay for it.